Good evening, everyone. So, welcome to week 11 tutorial session of post pathogen interaction. So, I'm Ananya Chatterjee. I'm a graduate student and a PMR fellow in IISc Bangalore, and I'm one of the teaching assistants for the course post pathogen interaction. Course instructor is Professor Himanshu Kumar from Department of Biological Science, IISER Bhopal. So, in today's class, we'll discuss the assignments of week 10. The first question is 1918 Spanish flu was result of infection by strain options H1N1, H5N1, H2N3, H1N2, H5N3. Answer is H1N1. Which one of the following is incorrect about influenza virus? Influenza is a positive sense virus. It has 8 segment genome. This is correct. It is a spherical to ovoid virus, so it is a polymorphic virus. Like it can be spherical, it can be ovoid, it can be like cylindrical. Okay. It has 4 major types. True, all are correct. So the question is, which of the following is incorrect about influenza virus? So influenza is a negative sense RNA. So incorrect is option 1. In the name of Sydney 0597 H3N2. 97 is strain number, HA number, NA number, year of isolation, virus subtype. So option year of isolation is the correct answer. Question number 4. The viral RNA isolated by Terence M. Tampe from Alaska permafrost was highly pathogenic avian influenza. This is the true. Was highly pathogenic swine influenza, was highly pathogenic civet influenza, was highly pathogenic bovine influenza. The answer is was highly pathogenic avian influenza. Chloroquine Chloroquine can inhibit influenza virus infection by disrupting the transport of viral particles, blocking fusion protein, increasing the pH, blocking M2 protein, and inhibiting viral polymerases. Chloroquine inhibits influenza virus infection by increasing the pH. Which of the following is a neuraminidase inhibitor? Amantadine, rimantadine, ammonium chloride, oseltamivir, and chloroquine. Answer is oseltamivir. Which of the following is not a mosquito-borne infection? Dengue is a mosquito-borne infection. Zika also mosquito-borne infection. West Nile as well. Japanese encephalitis as well. So the answer will be hepatitis C. Is not a mosquito-borne infection. Which of the following is a tick-borne infection? Hepatitis C, porcine virus infection, hepatitis B, hepatitis A, West Nile infection. So the answer is porcine virus infection. Which among the following is our arbovirus? Chikungunya virus, Zika virus, dengue virus. So 3 of them are arbovirus. Which among the following characteristics of the dengue virus are incorrect? Single-stranded positive sense RNA genome, segmented genome, cylindrical. So option 2 and 3 are incorrect. Option 1 is correct. It's a single-stranded positive stranded RNA present. It's not segmented genome, and it is also not cylindrical. It's a icosahedral, or you can say circular, but its proteins are icosahedrally arranged. So if you have any question, then we'll discuss, or we'll start with the parasite infection, example malaria. So different types of human parasite. Protozoa. Protozoa are unicellular eukaryotes. They replicate within the host cell based on means of locomotion. So we can so we can divide it based on different means of locomotion. So it can be flagellates. Example: Trypanosoma, Leishmania. Amoeba. Example: Entamoeba histolytica. Sporozoa, Toxoplasma gondii or Plasmodium species. Ciliates, Balantidium coli. Types of human parasite: helminths or worm organism. Those are multicellular, infect humans but can reproduce on own. Two phyla: Nematoda, that is roundworms, and Platyhelminthes, those are flatworms. Nematoda: Ascaris lumbricoides. Platyhelminthes: Schistosoma mansoni, Tinea solium. Common parasitic infections. Case fatality rate of common parasitic infections. So you can see very low rate of fatality rate in the case of schistosomiasis, guinea worm disease, malaria, echinococcosis and leishmaniasis. Moderate or high rate are there in leishmaniasis, sleeping sickness and echinococcosis. The case fatality rate is an epidemiologic matrix that measures measures the proportion of people who die from a specified disease among all positive cases over a period of time. This metric indicates the severity of a disease and is often used for prognosis. Common parasitic infections, average basic reproduction number are not of common parasitic infections. Infectious illness, leishmaniasis. So R naught is the average basic reproduction number of common parasitic infections. The average basic reproduction number R naught is an epidemiologic matrix that describes the transmissibility of infectious agents. R naught measures the expected number of secondary infections produced by a single infectious individual in a susceptible population during the mean infectious period. Malaria. So malaria caused by protozoan pathogens of the Plasmodium species. Plasmodium are single-celled eukaryotic organisms that belong to the phylum Epicomplexa, which is named for the apical complex that is involved in host cell invasion. Initial symptoms include fever, headache, shivering, and vomiting. If not treated early, depending on the species responsible for the disease, severe anemia, metabolic acidosis, heart failure, burden on kidney, or cerebral malaria may occur and even lead to death. Discovery of the malarial parasite in the gastrointestinal tract of the Anopheles mosquito led to the realization that malaria was transmitted by Anopheles. And Sir Ronald Ross received the Nobel Prize in Physiology in 1902. Malaria showed that malaria is caused by a single-celled organism, a protozoan protozoan of the Plasmodium family which attacks red blood cells. Leveran, so Charles Louis Alphonse Leveran also identified other single cell parasites that cause other diseases. Received the Nobel Prize in Physiology in 1907. Plasmodium species. 5 human infective Plasmodium species. Plasmodium falciparum causes the bulk of malaria associated morbidity. Plasmodium falciparum causes the bulk of malaria associated morbidity and mortality in sub-Saharan Africa. Plasmodium falciparum is associated with severe malaria and complications in pregnancy. Most malaria-related deaths are associated with this species. Plasmodium vivax accounts for the majority of malaria causes in Central and South America and in temperate climates. Plasmodium ovale found in Africa and Asia but is especially prevalent in West Africa. Plasmodium malari causes the mildest infections, although it has been associated with splenomegaly or renal damage upon chronic infection. Plasmodium knowlesi, initially considered as a parasite of non-human primates, can not only cause malaria in humans but can also lead to severe and even fatal malaria complications. Malaria endemic regions. So these are the color coded regions in the map. You can see the green coded are countries endemic in 2015. Yellow, this yellowish green is countries endemic to 2000. So this is the infectious cycle of the disease malaria, or you can say the plasmodium from CDC. So you can see mosquito takes a blood meal from one infected person. So it injects sporozoites. This sporozoites goes to liver cell, and in human liver stages they infect liver cell, and from there they form a seijohn phase. From there seijohn phase they gets ruptured, and merozoites are getting out. Then it enters into human blood stage. So immature trophozoid or ring stage to mature trophozoid to seijohn or gamocyte to ruptured seijohn, and then from the gamocytes it forms microgametes, and then the microgametes and macrogametes, and then those mosquito takes a blood meal and injects gametocytes. So microgamete entering macrogamete, exfagulated microgametocyte, eukaryote invade midgut wall, forms oocyst, and then it's a mosquito stage. So ruptured oocytes or release of sporozoites happens, and then when mosquito takes a blood meal, it again injects those sporozoites inside human. Life cycle of malaria parasites in mosquito. In the mosquito's gut, each male gametocyte produces 8 microgametes after 3 rounds of mitosis. The female gametocytes matures into a macrogamete. Male microgametes are motile forms with flagellate and seek the female macrogamete. The male and female gametocyte fuse, forming a diploid zygote which elongates into an eukaryote. This motile form exits from the lumen of the gut across the epithelium as an oocyst. Oocysts undergo cycles of replication and form sporozoites which move from the abdomen of the mosquito to the salivary glands. 7 to 10 days after the mosquito feeds on blood containing gametocytes, it may be armed and able to infect another human with Plasmodium species with her bite. Malaria symptoms: recurrent chills, fever and sweating. Symptoms peaks every 48 hours. Weakness, anemia, splenomegaly due to blockage of blood capillaries by merozoites. Damage several vital organs, cerebral damage, heart failure, burden on kidneys or renal failure. The symptoms of malaria is not due to Plasmodium species, but it may be due to the overproduction of TNF alpha, because cancer patients treated with TNF alpha show malaria-like symptoms. Host response to Plasmodium. Innate immunity provides the initial defense against infection when pathogens are first encountered, which is followed by the development of specific antibody secreting B lymphocytes. Children under the age of 14 has lowest immunity to malaria, as they produce low amount of antibodies against merozoites form of Plasmodium. It is believed that much of the pathology associated with malaria is a consequence of an imbalance in cytokine secretion, in which pro-inflammatory cytokine responses dominate. Toll-like receptor 9 or TLR9, a nucleotide sensing receptor, is involved in the host immune response against Plasmodium. TLR9 induced high levels of tumor necrosis factor production leads to clinical symptoms such as fever. In addition, the membrane of infected red blood cells stiffens, and this loss of deformability contributes to the obstruction of capillaries, which has life-threatening consequences in severe malaria when vital organs are affected. So, what are the drugs against Plasmodium? Aryl amino alcohol compounds, quinine, quinidine, chloroquine, amodiaquine, mefloquine, halofrantin, lumefantrine. These are different compounds. Antifolate compounds or antifols: pyrimethamine, proguanil, chlorproguanil, artemisinin compounds, artemisinin, dihydroartemisinin, etc. Chloroquine works by interfering with heme dimerization, the detoxifying biochemical process within the malaria parasite that typically yields malaria pigment or hemozoin. Artemisinin was first isolated from the stems, leaves, and flowers of Artemisia annua by Chinese scientists. Artemisinin effectively kills malaria parasites within host erythrocytes, after which dead parasites are killed by the spleen, leaving formerly infected red blood cells intact and circulating. Life cycle of malaria parasite in mosquito host.
Mosquitoes are usually vegetarian, preferring to drink nectar, fruit juices, and honeydew. Only a pregnant mosquito will bite humans, seeking nutrients from blood to nourish her developing eggs. If she drinks blood from someone infected with malaria, she too becomes infected with the disease. The tiny drop of blood filling the insect's stomach is teeming with malaria parasites. The parasite form that is deadly inside humans cannot survive in the mosquito's stomach and is slowly digested with the rest of her blood meal. However, back in the human host, a few of the parasites turned into a different type of cell, one that is sexual but remains dormant. Malaria sex is triggered when the warm human blood begins to cool inside the insect's stomach. The female form of the parasite matures into an egg. The male form takes a while longer to mature into sperm. This sperm is from an earlier feed. The fertilized cell can glide and begins to explore its new environment. It migrates to the outer lining of the mosquito's stomach before transforming into a cyst. Each cyst produces thousands of thin, tiny parasites which seek out and infest the mosquito's salivary glands. The next time this mosquito bites a victim, the malaria parasite will ride in with her saliva and infect another human. This year, 10% of people on Earth will be struck down with malaria. Most people who die from the disease will be pregnant women and children under the age of 5.
So, malaria vaccine candidates in clinical development. So, there are different stages of the pathogen in human as well as in mosquito. So in human you can say that it's a liver stage and a blood stage. So inhibition of different, we can target inhibiting different stages. So inhibition of merozoite invasion. So there are different drugs where are which are now at phase 1, phase 2. So these are the names of those drugs. And then prevention of IRBC mediated pathology, which is targeting blood stage. And again killing of infected hepatocytes which is targeting liver stage. Inhibition of sporozoite infection. Inhibition of sexual stage development, so which is targeting in mosquito stage. Mosquito with sporozoite gamma ray irradiation, transmission blocking vaccine. Now we will talk about parasitic infection, trypanosomiasis. It's also called African sleeping sickness. African trypanosomiasis, also known as sleeping sickness, is caused by microscopic parasites of the species Trypanosoma brucei. These are flagellated protozoa, causes chronic, debilitating disease characterized by its unique mitochondrial DNA, lives exclusively as an extracellular parasite. These are saliva type, salivarian, fecal contamination, or sterocoriarian. African sleeping sickness is transmitted by the tsetse fly or Glossina species, which is found only in sub-Saharan Africa. Two morphologically indistinguishable subspecies of the parasite cause distinct disease patterns in humans. Trypanosoma brucei gambians causes a slowly progressing African trypanosomiasis in Western and Central Africa. Trypanosoma brucei rhodesiensis causes a more acute African trypanosomiasis in Eastern and Southern Africa. African sleeping sickness. Clinically, sleeping sickness is divided into two stages. In the early stage, parasites can be found in the bloodstream and interstitial spaces of several organs. Patients experience myriad symptoms including chronic and intermittent fever, headache, pruritus, lymphadenopathy, and hepatosplenomegaly in the early stage. The parasite actively invades the central nervous system, marking the start of the late stage. Sleep disturbances and neuropsychiatric disorders dominate the clinical presentation, giving rise to the disease common name of sleeping sickness. Gambiancy infection is chronic with an estimated average duration of around 3 years of evenly divided between the two stages. Rhodesien's disease is usually acute and death occurs within weeks to months, possibly due to parasite being less adapted to humans. This is the life cycle of African trypanosomiasis or Trypanosoma brucei and Trypanosoma brucei rhodiense. So, tsetse fly takes a blood meal and injects metacyclic tri- trypomastigotes, and it goes to mammalian stage where injected metacyclic trypomastigotes transform into bloodstream trypomastigotes which are carried to other sites. Trypomastigotes multiply by binary fission in various body fluids, example blood, lymph and spinal fluid. Circulating trypomastigotes in blood during acute phase, usually undetectable in latent phase. And here cattle and possibly wild animals are the reservoirs. Tsetse fly takes a blood meal, bloodstream trypomastigotes are ingested. Bloodstream trypomastigotes transform into procyclic procyclic trypomastigotes multiply by binary fission. Procyclic trypomastigotes leave the midgut and transform into epimastigotes. Epimastigotes multiply in salivary gland. They transform into metacyclic trypomastigotes. Development of trypanosomes in the mammalian host and the tsetse flies. Slender cell proliferation, parasitemia, environmental sensing and density dependent differentiation, morphological and gene expression changes, cell patients. So morphological and gene expression changes, environmental sensing, survival in the vector, immune clearance and senescence, parasitemia, where again slender, transmission competence. Trypanosoma resistance to mammalian immunity. When the parasite is phagocytosed, stress-induced activation of adenyl cyclase of the trypanosome plasma membrane leads to the parasite releasing cyclic MP into myeloid cells and the consecutive activation of the protein kinase A and inhibition of TNF synthesis, thus preventing the immediate elimination of trypanosomes by the mammalian host. In the mammalian host, the whole parasite is covered with a coat of about 10 to the 7 identical molecules of glycoprotein, the VSG, which is anchored into the cell membrane by a glycolipid glycophosphatidylate inositol. Parasites are able to frequently switch their glycoprotein coat, continuously making the antibody response of the host obsolete and evading clearance. Antigenic variation in Trypanosoma brucei. So, duplication of VSG2 and translocation to expression site, it can happen. Duplication and translocation to expression site of VSG3. So, these are the antigenic variation. Drugs against Trypanosoma brucei, it is usually recommended that the trypanosomes should first be removed from blood and lymph by the use of suramine or pentamidine and subsequently from extracellular species CNS and CSF with melarsoprol. Other drugs include eflornithine and nifurtimox. Life cycle of trypanosoma in human host.
The life cycle of Trypanosoma cruzi in the human host. In this video, we see the life cycle of Trypanosoma cruzi in the human host. The trypanosome life cycle begins with the triatomine insect, which is infected with T. cruzi, biting a human host. Following the ingestion of blood, the metacyclic trypomastigotes are released with the feces from the insect near the area of the bite. Note that this form has great motility. Observe that the parasite enters the host through the bite site when the host scratches the site, or when the parasite moves to a region covered by mucosa. The parasite reaches the mammalian bloodstream and subsequently invades different cell types that it encounters, such as macrophages, muscle cells, epithelial cells, and neurons. In this example, the attachment of the trypomastigote form of the parasite to the macrophage surface is observed. The process of internalization via phagocytosis begins with the formation of pseudopods. During and after the internalization of the parasite, the parasitophorous vacuole assembles. Host cell lysosomes migrate toward the internalization region and fuse with the parasitophorous vacuole. The lysosomal content is released in the vacuole. However, the parasite is not affected. In the vacuole, the trypomastigote transforms into an amastigote. This transformation is accompanied by the disruption of the parasitophorous vacuole membrane. The amastigotes are released into the cytoplasm of the host cell and divide multiple times. Observe how many amastigotes are accumulating through the division process. They can occupy the entire cytoplasm of the host cell. Following division, the amastigotes transform into trypomastigotes, which show intense and constant movement that culminates with their bursting out of the cell. The trypomastigotes reach the extracellular space and, subsequently, the bloodstream. The parasites will now infect new cells.
موتناش الصاروخ بده يموتنا الجوع. احنا بالنسبة لنا في الخيم مش
In this video, we will learn development and life cycle of Trypanosoma cruzi in insect vector reduviate bugs. Trypanosoma cruzi is a parasite belonging to Hemiflagellates group. Development of Trypanosoma cruzi in invertebrate host, that is reduviate bugs, starts with ingestion of broader less motile trypomastigote forms that are present in blood of infected vertebrate human host. Are transmitted to reduviate bugs during the blood meal. A urocent trypomastigote forms transform into a mastigote forms in the foregut, that is stomach. Further, a mastigote transform into epimastigote and spheromastigote forms. Some intermediate form of parasites are destroyed in foregut. A urocent epimastigotes passes in gut further to the midgut or intestine. Epimastigotes forms multiply by binary fission and attaches to the gut epithelial lining. This attachment to the epithelial cells is due to presence of flagellum of epimastigote form. A urocent further the epimastigote form travel to the hindgut a urorectum. They finally transform into metacyclic trypomastigote forms in the hindgut. Metacyclic trypomastigote attaches to the cuticle lining of rectum and rectal sac of insect. When bug or insect passes feces or urine, these metacyclic trypomastigote forms are excreted in feces or urine. A urocent on subsequent insect bite metacyclic trypomastigote forms infects new vertebrate host and again starts life cycle in vertebrate host. This whole insect cycle takes about 15 days. That is called as extrinsic incubation period. There is no trans-ovarian transmission seen in the reduviate bugs, and once infected, they retain the infection throughout the life by the molting cycles.
The sleeping sickness cycle starts with a bite from a tsetse fly. If the fly bites someone who's already sick, it ingests parasites as well as blood. These parasites, called trypanosomes, accumulate in the fly's gut. 20 days later, they migrate to the tsetse fly's salivary glands and are ready to infect another victim when the fly next bites. The parasite enters the lymphatic system and then passes into the bloodstream and circulates through the body. At first, the host's immune defenses attack the parasite. To shield themselves, the parasites mutate, change form, and escape from the host's immune system until they're recognized and attacked by another wave of antibodies. This cycle of attack and defense is repeated several times. It's accompanied by symptoms such as intermittent fever, headaches, and joint pain, symptoms that mirror the inner struggle between the immune cells and the parasites. These first phase symptoms are sometimes too mild to be detected, but this is when it is actually easiest to treat the disease. If it isn't treated in time, the parasites advance to the brain. The blood-brain barrier is there, a filter to protect the brain against pathogens, toxins, and hormones circulating in the blood. But the trypanosomes breach it. At this stage, the disease is much more difficult to treat, as the blood-brain barrier also blocks some drug molecules. If the trypanosomes manage to enter into the brain, the sleep pattern becomes erratic. Infected people develop poor coordination and become confused, alternating between episodes of severe fatigue and agitation. Sleeping during the day, they can't sleep at night. The deterioration of the nervous system leads to coma. Left untreated, sleeping sickness is fatal.
ಅಗೋಡಾದಲ್ಲಿ ಸಿಗಲಿದೆ. Low rates on flights.
ಇಂಟರ್ನ್ಯಾಷನಲ್ ಮತ್ತು ಡೊಮೆಸ್ಟಿಕ್.
Hi, ever wished you could finish a book in a matter of minutes?
Hey everyone, in this lesson we're going to talk about the protozoal disease known as African trypanosomiasis. So to begin, what is African trypanosomiasis? Well, African trypanosomiasis is a disease caused by an infection with a parasitic protozoa from the genus Trypanosoma. And here's an image of those Trypanosoma parasitic protozoa. African trypanosomiasis is also known as African sleeping sickness, and we'll talk about why that is later on in the lesson. The Trypanosoma protozoa that causes African trypanosomiasis is transmitted by the tsetse flies of the genus Glossina, and here's an image of a tsetse fly. Now, it is estimated that about 100,000 deaths occur each year due to African trypanosomiasis. And the disease, African trypanosomiasis, is distributed in the sub-Saharan African region. Here's a map of the area where African trypanosomiasis occurs. And interestingly, this is also the distribution of the tsetse fly. So the tsetse fly, its natural distribution is in the same area, and it also transmits the disease African trypanosomiasis in this area. So what are some of the Trypanosoma species that cause African trypanosomiasis? Well, one of the species that causes African trypanosomiasis is Trypanosoma bruti rhodesiens. And Trypanosoma bruti rhodesiens causes an acute illness, and it is more located in East Africa, so in the eastern sub-Saharan African region. The second species that causes African trypanosomiasis is Trypanosoma bruti gambiens. And this species causes a more chronic condition. So as opposed to the bruti rhodesiens, which causes an acute and rapid illness, bruti gambiens causes a more chronic and slowly progressing illness. And an example of this is the meningoencephalitic stage. It takes about 250 days to occur, and we'll talk a bit more about these stages of the illness a little later. The Trypanosoma bruti gambien species occurs in the west and central part of the sub-Saharan African region, so as opposed to the bruti rhodesiens, which occurs in the East African region. Now, there are other species of Trypanosoma protozoa that cause African trypanosomiasis as well. Some of them include Trypanosoma bruti bruti, bruti lewisi, bruti congolens, and bruti evansi. So these also can cause African trypanosomiasis, but the major two are the most important species that cause African trypanosomiasis is the Trypanosoma bruti rhodesiens and Trypanosoma bruti gambiens. So we're going to talk about those two more specifically in this lesson. Now here's the life cycle of the Trypanosoma species that causes African trypanosomiasis. Now we mentioned that these Trypanosoma species are carried within this tsetse fly. Now the tsetse fly, as we mentioned before, is located within the sub-Saharan African region, and it is commonly found in warm and shaded areas, and the tsetse fly lives for about 1 to 6 months. When a tsetse fly encounters a person, that tsetse fly can take a blood meal or bite that person. And when they bite the person, they actually inject metacyclic trypamastogotes. Once these metacyclic trypamastogotes are inside the person, they can transform into bloodstream trypamastogotes. And then they can be carried to other sites in the body. So these trypamastogotes can then enter into other sites such as the lymphatic system, into lymph, and they can also even enter into spinal fluid. Once they are within these different areas of the body, the trypamastogotes can continually multiply by binary fission. And they can start causing a lot of the symptoms we'll discuss in the next couple of slides. Now, when trypamastogotes are inside the blood, if a tsetse fly encounters the person that is infected, the actual tsetse fly can actually take a blood meal and actually itself become infected with the bloodstream trypamastogotes. So the bloodstream trypanomastogotes can be ingested by the tsetse fly from an infected person, and then the tsetse fly can be a carrier of these trypanomastogotes that can infect other individuals later. So once the tsetse fly ingests bloodstream trypanomastogotes from an infected individual, these bloodstream trypanomastogotes inside the tsetse fly can then transform into procyclic trypanomastogotes within the tsetse fly's midgut. And then these procyclic trypanomastogotes can multiply again by binary fission. The procyclic trypanomastogotes eventually leave the midgut of the tsetse fly and then transform into epimastogotes. The epimastogotes can then enter into the salivary gland of the tsetse fly and multiply there. And then inside the salivary gland, they can then transform into the metacyclic trypanomastogotes. And those metacyclic trypanomastogotes can then infect another person when that tsetse fly bites someone else. So that is the entire cycle of the trypanosoma species that causes African trypanosomiasis. So if an individual is infected with a trypanosoma species, that individual does have some innate immunity through the protein apolipoprotein L1. And apolipoprotein L1 is in fact trypanolytic. So what happens is the trypanosoma species can engulf the apolipoprotein L1 through an endosome, but the apolipoprotein L1 actually causes a destruction of the trypanosoma. And with humoral immunity, there is a major variant surface glycoprotein, or VSG, on the trypanosoma organism. But what is interesting and what is important for the pathogenesis of trypanosomiasis is that the trypanosoma can actually periodically switch their VSG, or their variant surface glycoprotein, so they can make changes to that protein. So when the body of an infected individual makes antibodies to a specific variant surface glycoprotein, that trypanosoma can change or switch their VSG so that those antibodies are no longer effective. So they can continually avoid or attempt to avoid the infected person's humoral immunity response. And we call this antigenic variation. And this leads to nonspecific activation of humoral immunity. So as the body continually attempts to target that variant surface glycoprotein, the trypanosoma can switch the variant surface glycoprotein and evade the infected person's humoral immunity response.
I got a question for you. Do you have the courage to make 1 crore? क्या आप में वो जिगर है to make 1 crore?
Now, if an individual is infected with trypanosoma, species that causes African trypanosomiasis, what are some of the symptoms? Well, the symptoms are actually divided into 2 stages. Stage 1, early infection, and stage 2, late infection. So in an early infection, there are specific symptoms that occur. One of them that is most important is where the tsetse fly actually bit into the person, and it can actually cause what is called a trypanosomal chancre. And here is a trypanosomal chancre. This is typically, in most cases, the first sign that can be detected. It usually occurs 1 week after a bite from the tsetse fly. It's well circumscribed. It's described as rubbery, and it's considered painful, and it is indurated. Now, there's also a rash that can occur as well, a trypanosomal rash, and the rash typically occurs within 6 to 8 weeks after the initial infection. It is transient, urticarial, and it's macular. And the lymphatic system can be involved as well in the early stages of infection. So the trypanosoma travel to regional lymphatics. So once they are taken up into the body, they can travel into the lymphatic system in regional areas, and it can cause lymphadenitis, so an inflammation of the lymph nodes. With the Trypanosoma brucei gambiens, the lymph nodes in the posterior cervical area. The posterior cervical lymph nodes are the ones that are typically affected. And they lead to a painless and enlarged posterior cervical lymph nodes. And this is called Winterbottom sign. So here's an image of a Winterbottom sign. You can see here this individual has these enlarged lymph nodes in certain areas along their back and in their neck. Now the other species, Trypanosoma brucei rhodesians, has a different distribution with regards to the lymphatic system. It typically has less lymphadenopathy, so it doesn't affect the lymph nodes as much as Trypanosoma brucei gambians. But when it does, it affects the submandibular, axillary, and inguinal lymph nodes the most, as opposed to brucei gambians, which actually causes the posterior cervical lymph nodes to be affected. So now that we know what happens in the early stages of African trypanosomiasis infection, what happens in the later stages? Well, in stage 2, or the late infection of African trypanosomiasis, this is considered stage 2 or late infection once the central nervous system is involved. And once the central nervous system is involved, if left untreated, the mortality rate is 100%. The involvement of the CNS can be determined if there is an elevated white blood cell count within the cerebrospinal fluid, and it usually is about at least 5 cells or 5 white blood cells per microliter. With an infection of Trypanosoma brucei gambians, for stage 2 infection, during an earlier exposure, it can lead to progressive diffuse meningoencephalitis. It can lead to meningeal inflammatory infiltrates, and it can lead to widespread focal white matter demyelination. And with all of those effects, some of the symptoms involved due to an infection with Trypanosoma brucei gambians include headaches, difficulty concentrating, completing complex tasks, personality changes, psychosis, and tremors and ataxia. Other symptoms of a infection with Trypanosoma brucei gambians include alterations in circadian rhythm, leading to daytime somnolence or excessive daytime sleepiness. And now this is very important, as this is the reason why African trypanosomiasis is also referred to as African sleeping sickness, because of the alterations in circadian rhythm. And if left untreated, Trypanosoma brucei gambians infections eventually lead to a comatose state in patients. Now, an infection with Trypanosoma brucei rhodesians has similar symptoms to Trypanosoma brucei gambians, but has a more rapid onset, and it typically can take only weeks. So Trypanosoma brucei rhodesians infection is acute and severe and febrile, and it can also have symptoms of myocarditis and pericarditis. So how do we actually diagnose African trypanosomiasis? And once we diagnose, how do we actually treat it? Well, diagnosis of African trypanosomiasis can be accomplished by a blood smear to see if we actually can see the Trypanosoma protozoa. And a blood smear for diagnosing African trypanosomiasis is more important with Trypanosoma brucei rhodesians, because we can actually see them actually in the bloodstream or in the blood. And it can be positive in earlier stages, as Trypanosoma brucei rhodesians can rapidly infect and rapidly proliferate in a patient. And to detect on a blood smear, typically we need to see about at least 5,000 protozoa per ml of blood. Now, with tissue aspirate, this can be important, as if the Trypanosoma species infects the lymph nodes, we can actually take an aspirate of the lymph node and look at it under a microscope, and then we might be able to see the Trypanosoma species that way. And this is important with the Trypanosoma brucei gambians, as these the species typically involves the lymph nodes. So a tissue aspirate can be important for diagnosing patients with an infection with Trypanosoma brucei gambians. As well, cerebrospinal fluid So a spinal tap is important to see if we can detect any trypanosoma within the spinal fluid, or if we can see any white blood cells. So we may be able to see pleocytosis within the cerebrospinal fluid. Serology is also important to detect, to see if we can find any antibodies to any of these trypanosoma species. And PCR is also a method to diagnose patients with infections. One of the most important things before we start treatment is to check the patient's cerebrospinal fluid. That's one of the first things we want to do, because that will determine if it's an early stage or stage 1 infection, or if it's later stage or stage 2 infection. And that's important because depending on the stage, if it's early or late, that will change what treatment we use. So if the patient has been found to be infected with Trypanosoma brucei gambians, and it's been determined that it's an early infection, then we can use pentamidine. And if we don't use pentamidine, we could use suramin. And if it's been found that it is a late infection of Trypanosoma brucei gambians, we would have to use eflornithine plus or minus nifurtimox. So the reason that we change the medication, the reason that we use eflornithine as opposed to pentamidine, is because pentamidine does not cross the blood-brain barrier well. Eflornithine does. So in order to treat a CNS-involved infection of trypanosomiasis, we'd have to use something that can cross the blood-brain barrier. So eflornithine would be the one we'd use. Now, if it was found that the patient was infected with Trypanosoma brucei rhodesians, and it was determined that it was an early infection, that there was no CNS involvement, we could use suramin as the treatment. But if it was found to be a late infection, CNS was involved, and it was a Trypanosoma brucei rhodesians infection, we'd have to use melarsoprol. And you might be wondering, okay, why can't we use eflornithine in these cases? Well, it appears that eflornithine has no effect on treating Trypanosoma brucei rhodesians, so we have to use melarsoprol. So that's the reason why it's important to also figure out what species we're dealing with as well. And melarsoprol can be 10 daily injections can be used to treat, and that's been shown to be effective. Anyways, guys, that was a lesson on African trypanosomia.